We found potent binders and confirmed them by docking. But here's the hard truth of drug discovery: a potent molecule is not a drug. Most candidates die later, not because they don't bind, but because of ADMET, how the body absorbs, distributes, metabolizes, excretes, and is poisoned by them. Today we score those liabilities in code, and then do the central move of medicinal chemistry: fix 1. ADMET stands for absorption, distribution, metabolism, excretion, and toxicity, everything that happens after a molecule enters the body. We can't simulate all of it, but 3 fast, codable filters catch a lot early. QED, a drug likeness score from 0 to 1. SA score, how hard the molecule is to synthesize. And Brenk alerts, a catalog of reactive and toxic substructures you simply want to avoid. So we write one profiling function, QED, synthetic accessibility, and any Brenk alerts, straight from the structure. Run it across a few molecules and you get an instant scorecard. Notice the real drugs, gefitinib and erlotinib, carry only mild flags from their long flexible tails. This is the kind of table a medicinal chemist scans before deciding what's worth making. Now optimization. Here's a hypothetical hit on our quinazoline scaffold, but someone hung a nitro group on it, and nitro aromatics are a classic mutagenicity flag. The profiler catches it immediately. So we make the medicinal chemist's move: swap the nitro for a benign chlorine, keep everything that binds, and rescore. The alert vanishes. Look at the before and after. One group swapped, nitro out, chloro in, and the toxic alert is gone. The drug likeness score jumps from 0.57 to 0.79, and the molecule actually gets easier to synthesize. We kept the part that binds and removed the part that would have killed the program. Real campaigns repeat this loop hundreds of times, trading off potency against ADMET at every step. So we scored the liabilities that sink most drug candidates, and watched a single, targeted edit turn a flagged hit into a clean, more drug-like molecule. That's optimization, the daily work of medicinal chemistry, in a few lines of RDKit. Next episode is the capstone. We chain everything, from ChEMBL data to a ranked, filtered, docked shortlist, into one reproducible mini-campaign against EGFR. See you at the finish line.